Welcome to this explainer. Have you ever just stopped for a second, maybe while you're holding a cup of coffee or watching someone sprint down the street, and wondered how that movement actually happens? I mean, we live our entire lives on this big macroscopic scale, but every single everyday movement we make is actually being driven by an entirely invisible, highly coordinated microscopic universe. And today, we're going to bridge that gap. Let's dive right in with something so incredibly common you literally don't even think about it: blinking. Imagine a speck of dust blows right toward your face. Almost instantly, this thin little flap of your eyelid snaps down to clear it away, and then pops right back up. It feels instantaneous, right? Super simple. But to answer how that simple blink actually happens, we have to look way past the eyelid itself. We really need to zoom deep, deep into the body, past the nerves, past the gross muscles, all the way down to the specific molecules and ions that are physically interacting to create that exact split-second movement. Now, we all know the golden rule of biology here. Anatomy is the what, and physiology is the how. Form strictly dictates function. A structure can only do its specific job because of its exact physical shape. But to really grasp how these structures support life, we can't just stop at macroscopic bones and muscles. We have to realize that this absolutely unbreakable rule of form and function goes all the way down to the cellular and, more importantly, the molecular level. And that brings us to the ultimate goal of all this functioning: homeostasis. Your body is basically working relentlessly, 24/7, to maintain this steady state of internal balance. If your internal temperature spikes even a little, your physiology actively fights back to restore it. But here's the thing: homeostasis isn't just some broad textbook concept. It is an active, incredibly demanding process that requires perfect coordination, and it's driven entirely by invisible molecular machinery that never clocks out. Okay, so to pull off this massive homeostatic balancing act, the body relies on a very strict hierarchy. We've got the broad organ systems, which are built from specific organs, right? And those are woven from tissues, which are fundamentally made of cells. But the true foundation of all of it? Molecules. To really decode human physiology, we absolutely must trace function all the way down to this molecular baseline. Let's use muscle tissue as our case study to trace this journey from the macro down to the micro. When you think of muscles, you're probably just thinking of locomotion: walking, running, moving around. But muscle physiology does so much more to maintain homeostasis. It powers peristalsis in your digestive tract, it controls the constriction of your blood vessels, and get this, here's something truly wild: about 75% of the ATP energy used during muscle contraction is actually released as heat. So when you shiver to stay warm, that is a prime example of muscle physiology working right at the molecular level just to maintain your core body temperature. Looking at the big picture macroanatomy, a skeletal muscle isn't just one big solid slab of tissue. It's highly, highly organized. The whole muscle organ is built out of these discrete bundles called fascicles, and those break down even further into elongated muscle fibers. Its physical form is just perfectly designed to distribute force. But if we want to find out where that actual pulling force originates, we've got to go smaller, much smaller. To really find the source of that pulling force, we have to cross a major boundary. Leaving gross anatomy in the rearview mirror, histology and cytology reveal that what looks like simple bundles of meat to the naked eye are actually these highly specialized, incredibly complex cellular machines designed for one specific microscopic function: contraction. Inside the muscle cell, or myocyte, things get really interesting. We find specialized features unlike anywhere else in the body. They have this thing called the sarcoplasmic reticulum, which acts like a rapid-release intracellular vault for calcium ions. They have transverse, or T-tubules, that relay electrical action potentials deep into the core of the cell. And most importantly, they are completely packed with myofibrils. That's the actual contractile apparatus spanning the entire length of the muscle fiber. Now, if we take just one of those myofibrils and look even closer, we finally hit the absolute fundamental unit of muscle physiology: the sarcomere. This is the exact microscopic scaffolding where muscle shortening physically occurs. Your bicep doesn't contract as one giant piece. Instead, millions of these tiny sarcomeres lined up end to end shorten all at the exact same time. This right here is where molecular physiology actually translates into macroscopic movement. By zooming in on the banded, sort of striped appearance of a single myofibril, we can strip away the cell membrane entirely, and what's left? The pure molecular structure of a single sarcomere. Basically, it's just a beautifully organized scaffolding of overlapping parallel protein filaments. At this purely molecular level, the entire magic of movement comes down to two primary proteins. You have the thin filaments, made mostly of a protein called actin, and the thick filaments, made of myosin. They lay perfectly parallel to one another, overlapping in this really precise pattern. Remember that golden rule about form dictating function? Well, it is absolutely fascinating to realize that the specific, three-dimensional physical shape of just these two tiny protein molecules is exactly what allows you to blink, walk, or even breathe.
Which perfectly illustrates the sliding filament theory. For a long time, researchers were scratching their heads. How do muscles actually get shorter? Did the proteins themselves shrink? Did they sort of fold up like an accordion? Well, turns out, the actin and myosin filaments don't change their actual length at all. Instead, they literally slide past one another. The thick myosin grabs the thin actin and essentially pulls it toward the center. And since the actin is anchored to the ends of the sarcomere, the sliding action physically shortens the entire unit.
So, under the right conditions, specifically when that calcium is released from the sarcoplasmic reticulum vault we mentioned earlier, these little heads sticking off the thick myosin filaments reach up and physically attach to the actin, forming what's called a cross bridge. The myosin head then bends, pulls the actin filament, detaches, and resets. This repeated grabbing and pulling is called cross-bridge cycling. It's like millions of molecular ratchets working in perfect unison to create a sliding movement that ripples all the way up to create a massive muscle contraction. This brings us to such a profound realization about human biology, and our sources summarize it perfectly. Even the three-dimensional structure of certain molecules is essential to their function. Just think about the journey we just took. You blink your eye. The eyelid muscle contracts. The fascicles shorten, driven by the fibers, driven by the myofibrils. But at the very bottom of that chain, that macroscopic blink only happened because the precise three-dimensional shape of a microscopic myosin molecule allowed it to grab an actin molecule and pull. Your entire macroscopic life relies 100% on invisible microscopic physiology. So the next time you take a step, take a deep breath, or even just casually blink your eyes, maybe pause for a second. You aren't just casually moving a limb. You are activating an unfathomably complex, highly organized cascade of biological events, from the broad organ systems all the way down to the intricate dance of overlapping protein filaments. Form strictly dictates function just to keep you alive and moving. It really makes you wonder, what other incredible, invisible molecular machinery is working inside you at this exact second? Thank you so much for joining me in this explainer. Keep learning, keep wondering, and I'll catch you next time.